The study of genetics reveals the intricate mechanisms that determine how traits are inherited and expressed in living organisms. By understanding the principles of inheritance, scientists and students alike can predict the outcomes of genetic combinations, whether they follow the classic patterns described by Gregor Mendel or the more complex variations seen in non-Mendelian genetics. Today, we'll explore how to use monohybrid and dihybrid crosses to predict genetic outcomes, including the inheritance of traits influenced by incomplete dominance, codominance, sex-linked traits, and multiple alleles. Let's begin with the foundational principles established by Gregor Mendel, often referred to as the father of genetics. Mendel's experiments with pea plants demonstrated that traits are passed from parents to offspring in predictable patterns. He observed that each trait, such as flower color or seed shape, is controlled by two alleles, one inherited from each parent. In Mendelian inheritance, one allele is dominant, meaning it masks the effect of the other allele, which is recessive. For example, if R represents the dominant allele for round seeds and R represents the recessive allele for wrinkled seeds, a plant with the genotype RR or RR will have round seeds, while only RR will produce wrinkled seeds. A monohybrid cross examines a single trait. To predict the outcomes of a monohybrid cross, we use a Punnett square, a diagram that shows the possible combinations of alleles from two parents. For example, if two heterozygous plants, RR, are crossed, the Punnett square reveals that their offspring have a 1:2:1 genotype ratio, 25% RR, 50% R, and 25% RR, and a 3:1 phenotype ratio, with 75% round seeds and 25% wrinkled seeds. A dihybrid cross, on the other hand, examines two traits simultaneously. For instance, consider seed shape, round R or wrinkled R, and seed color, yellow Y or green Y. If two heterozygous plants, RY, are crossed, a Punnett square with 16 squares is used to predict the offspring's genotypes and phenotypes. The results show a 9:3:3:1 phenotype ratio, 9 with round yellow seeds, 3 with round green seeds, 3 with wrinkled yellow seeds, and 1 with wrinkled green seeds. While Mendel's principles provide a strong foundation, not all traits follow these simple, dominant recessive patterns. Let's explore some non-Mendelian inheritance patterns. First, consider incomplete dominance, where neither allele is completely dominant over the other. Instead, the heterozygous genotype results in a blending of traits. A classic example is flower color in snapdragons. If R represents the allele for red flowers and W represents the allele for white flowers, a plant with the genotype RW will produce pink flowers, a mixture of red and white. A cross between two pink flowers, RW crossed with RW, results in a 1:2:1 phenotype ratio, 25% red, 50% pink, and 25% white. Next is codominance, where both alleles are fully expressed in the phenotype of the heterozygote. A well-known example is the inheritance of coat color in certain cattle breeds. If R represents the allele for red coat color and W represents the allele for white, an animal with the genotype RW will have a roan coat with both red and white hairs equally expressed. A cross between two roan cattle, RW crossed with RW, results in offspring with red roan and white coats in a 1:2:1 ratio. Another fascinating pattern is seen in sex-linked traits, which are associated with genes located on the sex chromosomes. In humans, females have two X chromosomes, XX, while males have one X and one Y chromosome, XY. Traits linked to the X chromosome, such as colorblindness, exhibit unique inheritance patterns. For example, the allele for normal vision, XN, is dominant, while the allele for colorblindness, XN, is recessive. A female carrier, XNXN, has normal vision but can pass the colorblind allele to her offspring. If a carrier female is crossed with a male with normal vision, XNY, the offspring have a 50% chance of inheriting normal vision for both sexes, a 25% chance of a colorblind male, and a 25% chance of a carrier female. Importantly, because males inherit only one X chromosome, they are more likely to express sex-linked recessive traits. The concept of multiple alleles adds another layer of complexity. Some traits are controlled by more than 2 alleles, although each individual still inherits only 2 alleles. A classic example is the ABO blood group system in humans, determined by 3 alleles, IA, IB, and I. The IA and IB alleles are codominant, while I is recessive. Using this system, individuals with the genotype IIA or III have type A blood, those with IBIB or IBI have type B, those with IAIB have type AB, showing codominance, and those with IA2 have type O. Predicting the outcomes of blood type crosses involves using a Punnett square to determine the possible combinations of these alleles in offspring. The ability to predict genetic outcomes has profound implications in fields such as medicine, agriculture, and conservation. For example, genetic counseling uses these principles to help families understand the risks of inherited disorders. In agriculture, breeders use knowledge of inheritance to develop crops and livestock with desirable traits, such as higher yield or disease resistance. In conclusion, the principles of inheritance, whether Mendelian or non-Mendelian, provide a roadmap for understanding how traits are passed from one generation to the next. Monohybrid and dihybrid crosses reveal the predictable patterns of Mendelian traits, while the study of incomplete dominance, codominance, sex-linked traits, and multiple alleles uncovers the complexity of genetic variation. By analyzing these patterns, we not only unlock the mysteries of heredity, but also harness this knowledge to solve real-world challenges and improve the quality of life.